We're going to start off by looking at the structure of a skeletal muscle. You know those dolls, the nesting dolls, where like you open one up and there's another inside it, and you open another and there's another inside there, and so on? That's what muscles are like. The basic structure of a muscle is it's a bunch of tubes inside of tubes. So if we start off with an individual cell, which remember, a muscle cell is called a fiber. Here is a fiber, which is just one cell. And skeletal muscle cells are long. They're not the spherical typical cell you see. They're long tubes, and they have long tubes of protein inside them. And the two main strands of protein in here are actin and myosin. Each fiber has a cell membrane called the sarcolemma. So every cell has a membrane. You've already learned about cell membranes in a previous chapter. When you're talking about a muscle cell, you give that membrane a special name. Sarcolemma is just the membrane of a muscle cell. These cells are then wrapped in another layer of tissue called endomysium. Okay, now for that whole nesting doll part. If you take a group of fibers and you wrap them up together in perimysium, then you have a fascicle. So this structure is a fascicle. Here's another fascicle. See how you have kind of these groups in a muscle? Each group is a fascicle. Then when you put those groups together, you get your entire muscle. And this muscle is wrapped in epimysium. So a muscle is a group of fascicles, a fascicle is a group of fibers, a fiber is a group of actin and myosin strands. Okay. Here we're looking at it sideways. So we're taking this picture and we're turning to look at it sideways. So here we're kind of looking at it head on. If we take this section right here, this fiber, and we turn it to look at it sideways like this. That's what this picture is. It's a side view of a fiber. So this is the fiber. And remember I said you have the actin and myosin inside of there. So you can see tubes inside the fiber. This is where our actin and myosin is. We can take these tubes, which are called myofibrils, and we can divide them up into units. Notice you have these kind of red, well no, sorry, here, these zigzag lines here. Each zigzag line is a Z-disc. Because it zigzags like a letter Z, so the name makes sense. From one Z-disc to the next is a unit called a sarcomere. So if we look at this picture up here, there's the borders of a sarcomere. there's another one, there's another one, there's another one. So like here's 1, 2, 3, 4, 5, 6, 7, 8, 9. There's 9 sarcomeres shown there. Okay, here we're looking up close at one of them. Within the sarcomere are the actin and myosin. So in this picture, and different books use different color coding schemes. In this picture, the blue lines here are the actin, and the red lines are the myosin. Notice the actin is a thin line, the myosin is a thick line. So actin is also called the thin filament, and myosin is also called the thick filament. Okay, so now this picture down below is an up close. So now we're looking even more up close. Here you can see, here again is the actin. Here is the myosin. And this is a Z disc. Here's another Z disc. So you have Z disc on the ends. Okay, then there's a structure in the middle here called the M line. So muscles contract. We take a minute, let's go back and kind of think of the big picture here. We know that muscles contract. That's the main feature of a muscle. Contraction means that it gets shorter. That's how muscles produce movement. When you contract a muscle, it gets shorter, and your muscles are attached to your bones. So when it gets shorter, it pulls on a bone and it makes that bone move. And that's how your body moves. So we're looking in extreme detail at the microscopic level of how a muscle contracts so that it can pull on a bone and make your body move. The basic idea here, you have at the M line, you have this empty space. So you kind of have this gap here. If you can get this actin to go in this way, and this actin to go in this way so that they meet at the M line, the actins, notice, are attached to the Z disc. So when they move in, they will pull the Z disc in. Z disc will come in closer to the M line, and this whole unit will be shorter. Here's that same picture again. This is a muscle that's relaxed. Notice you have this nice big gap here, this open space. Here it is when it's contracted. See how the actins have moved in? They're meeting at the M line. There's no more open space there. And if you compare the length of these, look at where your Z disc are here. Look at this length. Look at where your Z disc are here. Look at this length. See how the second one is shorter? That's a muscle contraction. So the basics of a muscle contraction, if we put it in the simplest form possible, is that the actins meet at the M line, and that pulls the Z disc in, and that makes the sarcomere shorter. So the actins from opposite sides move toward each other. To meet at the M line. That they are attached to the Z disc, so when they move in, they pull the Z disc closer to the M line. And when the Z disc are closer to the M line, the sarcomere is shorter. and that is a contraction. A contraction is when the sarcomere gets shorter. And remember back up here how you have all these sarcomeres lined up end to end? If each one of those gets a little bit shorter, that whole fiber gets shorter. And when you have all your fibers get shorter, your whole muscle gets shorter. And here you can see, here's the bone. When that muscle gets shorter, it's going to pull that bone and make it move. So now let's go into a little more detail of how this happens. The actins can't just magically move. Something has to make them move. That's where the myosin comes in. The myosin has all these heads. See all these little bumps here? These are myosin heads. The myosin heads are going to grab the actin and pull it in. Think about if you're out on a boat and you're pulling in the anchor. The rope would be the actin. Your hands grabbing that rope and pulling it in would be the myosin heads. So the myosin heads are going to attach to the actin, and this is called cross bridge formation. So whenever a myosin head is attached to the actin, that's called a cross bridge. So if you're thinking of yourself in a boat, whenever your hand grabs the rope, you formed a cross bridge. Then the myosin heads bend. And when they bend, that is called power stroke. And by bending, they pull the actins toward the M line. So the power stroke would be like you pulling your arm in towards yourself in order to pull that rope in towards yourself. So all these little myosin heads, they're going to come up. A myosin head will attach like this, then it bends this way, and that's what pulls the actin in. And over here, they'll attach like this, and then they'll bend this way and pull the actin in. And the actins move toward the M line. Now, you have to have control of this. So your brain decides when you want to move your body. So it's deciding when you're going to contract your muscles. The way you control this is by controlling when you allow cross bridges to form. So if it were up to myosin, it would always do this. Myosin always wants to cross bridge and power stroke. Every time it does, your muscle contracts. Well, you don't just sit there all the time with all your muscles contracted. They have to contract and relax alternately. If they just stayed contracted, your body would just be locked up. So you have to be able to stop this from happening when you want your muscle to relax. The way you do that is with a couple more proteins called troponin and tropomyosin. So if we look at this picture, this is even more up close. Here's the actin. Here's the myosin. Right? And here's the myosin head. The myosin head is the part that's going to attach to the actin. You have these other proteins here that are blocking. Tropomyosin is another protein in muscle. It blocks the myosin head from binding to the actin. So this is showing a muscle that is relaxed. When a muscle is relaxed, the myosin heads can't bind to the actin because tropomyosin is in the way. Then when you want to contract your muscle, so now let's say your brain wants to contract muscle. In order to do that, you have to move the tropomyosin. Well, this is where calcium comes in. Remember, the sarcoplasmic reticulum is a structure in a muscle cell that stores calcium. So when your brain wants to contract a muscle, you're going to send a nerve signal. So you're going to have a signal that is going to start in your brain. It's going to travel along a nerve to your muscle. You're then going to secrete acetylcholine. Acetylcholine is a neurotransmitter onto the muscle. The acetylcholine will cause what's called an action potential, which just means it's the electric signal. So the acetylcholine will cause an action potential on the sarcolemma of the muscle cell. That action potential will go down the T-tubule. The T-tubule is a tunnel that just carries that electric signal from the surface down into the muscle fiber. So you have an action potential on the surface of the muscle fiber. That then goes down the T-tubule, this little tunnel, into the fiber. And it stimulates the sarcoplasmic reticulum to release calcium. The calcium comes out here. Remember, we have tropomyosin preventing the myosin head from binding to actin. These red dots here are calcium. The calcium binds to troponin. When it does, the troponin then moves the tropomyosin. So that's what you can see happening in the middle picture here. Here is the calcium binding the troponin, and when it does, it moves the tropomyosin out of the way. So see this little gap here in the actin? See how that's perfectly shaped for the myosin head to fit in? And these arrows here, these black arrows, are showing you that the tropomyosin used to be there in that little gap. Now it has moved out of the way. Once it has moved out of the way, now the myosin heads can attach. So now we can have cross-bridge formation. So once the tropomyosin is moved out of the way, you can have cross-bridge formation. Remember, cross-bridge formation means the myosin heads are attaching to the actin. Then in the next picture is power stroke. So notice the myosin head. See how it attaches like this? Then it bends like this. That's power stroke. And notice your actins move in. This one attaches at this angle, then it bends to this angle. It pulls the actin this way. So then you have your power strokes and the actins are pulled toward the M line. And the actins are attached to the Z disc, so then they pull the Z disc to the M line, or closer to the M line. And then it is shorter, which is the muscle contracted. So if we put this all together, this picture, there's the sarcomere relaxed. You would have tropomyosin blocking the myosin. Notice that the myosin heads are not attached to the actin in this picture. The tropomyosin is blocking it. So this picture doesn't have the tropomyosin, but if we drew it in, it would be like right here. There'd be the tropomyosin is blocking the myosin from binding to actin. Then you get a nerve signal from your brain, goes through a nerve, The neuron secretes acetylcholine onto the muscle. That causes an action potential on the sarcolemma. That action potential goes down the T-tubule. It stimulates the sarcoplasmic reticulum to release calcium. The calcium binds to troponin, which moves tropomyosin out of the way. Now you're free to form cross bridges and power stroke. And then you get this, the contracted fiber, where the myosin heads did attach to the actin, pull it in toward the M line. Now the muscle is shorter, it has pulled on a bone, the bone has moved. That's how you produce your body movements. We're going to start off with the basic structure of a muscle. In a muscle, at the most microscopic level, you have these long strands of protein. The two main ones are actin and myosin. So if we pretend that these red and yellow pipe cleaners are actin and myosin, this is a representation of the inside of a muscle fiber. The muscle fiber has a cell membrane called a sarcolemma. So this pink and black cloth represents that. So muscle has long strands of protein, the actin and myosin, and that is wrapped up in the sarcolemma, the cell membrane. Then you take a group of those and you wrap those up together. So individual fibers are wrapped up in endomysium, so you would have a layer of endomysium outside of this. Then you take several fibers. So here you can see individual fibers. The pink represents the individual fibers, each wrapped up in its own sarcolemma and endomysium, and you wrap those up together in perimysium. So the green represents perimysium. Now you have a bundle of muscle fibers in perimysium. This is a fascicle. Then, if you take several fascicles, so each bundle of green here represents a fascicle, and you wrap those up together in another layer of connective tissue called epimysium, then you have an entire muscle. So this bundle represents an entire muscle. It has epimysium, which is the tan. Inside of it, you have these units called fascicles, represented by the green. The outer layer of the fascicle is perimysium, which is this green sheet. If you unroll one of those, you find within it individual muscle fibers, the pink, each wrapped up in endomysium. Now let's come over here and look at these models. This is a model of a muscle fiber. So this is showing us a cell of a muscle. Remember, fiber is another name for a cell. Within it, you have units called sarcomeres. So from one red line here to another red line is a sarcomere. And you can see how these all line up end to end. In your book, it describes it like a train, how they're all lined up end to end, are the sarcomeres. The blue part here is the sarcoplasmic reticulum. That's where the calcium is stored. And this purple is the T-tubule, and here's your sarcolemma, the cell membrane. So the cell membrane will receive a signal from a neuron. The neuron's going to secrete acetylcholine onto here. That'll cause an action potential on the sarcolemma. The action potential, which is an electric signal, will travel down the T-tubule. That'll stimulate the sarcoplasmic reticulum to release calcium. The calcium will come out here, and it will bind to troponin, move tropomyosin. The actin and myosin can form cross bridges. And these red lines are the Z-disc. They will get pulled closer together. And if each one of these things, each unit here, each sarcomere, has its Z-disc pull in toward the M line, the sarcomere gets shorter. And when all of these get shorter, the whole fiber gets shorter. And that's a muscle contraction. So this model is showing us an individual fiber. It would be like what we have here in the pink with the actin and myosin inside. We're looking at that up close. These green lines are the myosin. The purple lines here are the actin. This model is a sarcomere. So we're taking here what we have from one Z disc to the next, this individual sarcomere, where you have hundreds of them lined up end to end in a muscle, and we're looking at it up close. So the yellow lines here represent the T-tubules. That's what carries the action potential down into the muscle fiber. The teal is the sarcoplasmic reticulum. That's where your calcium is. We take this off, we can look in here. Here's the M line, the center of the sarcomere. Here's a Z disc, here's another Z disc. That's the ends that are going to get pulled in closer together. The blue here is the myosin, and you can see the myosin heads. The red is the actin. The yellow stripe is the tropomyosin, and the green is the troponin. So when you get that electric signal from the neuron, so the neuron secretes acetylcholine onto the muscle, that causes an action potential in the sarcolemma. The action potential comes down the T-tubule. When you have an action potential in the T-tubule, the sarcoplasmic reticulum releases its calcium. That calcium diffuses out and it binds to the troponins, the green things here. Once the calcium binds to the troponin, it moves the tropomyosin, the yellow stripe, out of the way. Notice here the myosin heads. See how they want to come up and connect to the actin. As long as tropomyosin is in the way, they can't connect to the actin. Once the calcium binds to troponin and moves tropomyosin out of the way, now the myosin heads are free to bind to actin. So that's what happens, and that's called cross-bridge formation. The myosin heads bind to the actin. Same thing will happen on this side. The myosin heads will bind to the actin. Then you have power stroke. These myosin heads bend. So they can stand up and they can bend forward. So once you get the cross bridges, the myosin heads bend and that pulls the actin in. Think about like pulling in an anchor on a boat. You have to keep grabbing the rope and pulling over and over. So then the myosin heads will let go, they'll pop back up, they'll bind, and then they'll bend again. Each time they do that, they pull the actin in a little bit. So the actin gets pulled in little by little toward the M line. Same thing happens over here. It gets pulled in little by little to the M line. Then you have your sarcomere shorter. So when you started out, the sarcomere was like this. You had this gap here, this open space. Notice your Z disc here, your Z disc here. Look at the length of the sarcomere. The calcium binds troponin, moves tropomyosin. You have cross bridges form. Then the myosin heads power stroke. This one gets pulled this direction. This one gets pulled this direction. The Z disc are now closer to the M line. The sarcomere is shorter, and that's how a muscle contracts.